Good evening, everybody. Welcome to Taxonomy Tuesday. My name is Chris. I'll be your host. I have a short video I made about what it is we do with some of these things that we find. And I'm not sure if this is a thing that anyone will be really interested in or not. Uh, there's no audio, so I'm going to talk over it. And if you have something to add, jump in. Afterwards, we'll do some member finds. So if you have something you want to share from your machine, just put it in the chat and we'll try to get to everybody in a orderly fashion. So let me share my screen and start going. Well, there we go. So I found these around the corner, the edge of the school property. Slightly decurrent gills, a little brownish, creamy colored cap. I laid it on a slide on my little contrast piece of plastic, little black, little white, and I put it in the sun. So that spore print is a good color, that creamy tan color. And then, after you get some of these things and you let them sit around and get a spore print, I like to reuse my slides. So there you go. That's the brand of little alcohol wipes. I think people just get them for their glasses. So after I wash off my slides, they might have some little water spots or something. So I give everything a good wipe, and it kind of stays wet a little bit. So like you could see, I don't put them directly down right away. I'll perch them on something, uh, and sometimes after the spore print like this one there, it's a little dried out. This is the next day I got to this, so the material's a little tougher, and it's a little less slicing and a little more scraping. Uh, Sometimes just trying to get some little chunks of the gill, and I'll transfer these little by little, but I'll just kind of make a pile of stuff to work from when they're a little bit dried up like this. I left the little ruler there next to it just to kind of, for piece size, which I went a little big on these. After they're not, as fresh as they could be, I'm just looking for stuff. And if I don't find anything right away, sometimes I could go back and just take some more pieces and start over, you know, go back to it. A lot of times I'll make more than one of these, uh, one with water, and then another one I'll do with some dye or whatever. It's nice to have the toothpick out there to just kind of knock pieces off where you want them. I like putting a couple on a slide at once because then it just gives you more to look at, under one preparation. If you're not finding anything on one piece, maybe you find something on another one. Uh, After you get a little bit of water in there, you can see it's just sitting kind of to one side. So after you squish it down, it'll spread that out a little bit. And you got to watch to not get suction on the next one. Otherwise, you'll do what I did here and take it with it, the slide that I used to crush it with. And then I'll just, dry up any excess water or whatever so you're not getting it all over your microscope objectives. And after you're pleased with whatever you got, you can go ahead and try to get as much evenly spread liquid under there so you don't have too many air pockets, which will show up as you're looking for stuff as it's like a big black line or something like that where the water and the air meet, and that'll prohibit you from seeing anything decent. Sometimes takes a couple tries to get everything just the way you want it. It's worth taking your time and just doing this. I was doing this at my dining room table, so wherever's comfortable to make a little spot so you can work comfortably and not have to put stuff all over around your microscope, which I don't know about anybody else, but you don't want too much clutter around when you're trying to do the next bit of stuff. Also, I like just working on either like an all white surface like this, so you can really see what you have and what you don't. It's easy to keep track of, and the smaller the pieces get, the harder it is to keep track of anything. And then even like on the second one that I'm going to put some dye on, I like starting with water at least when I'm preparing it because I don't want too much dye. I like putting a little bit on the side and letting the water pressure kind of just take it in a little bit. So instead of filling back in with water, I'll start with dye, and then this was Congo red, and it'll just shoot right underneath there, and then I'll dilute it out and draw it from one side to the other so it kind of goes around and hits all the pieces, and just kind of bleed it from one side to the other because you don't want too much dye under there either. It gets hard to see when your whole screen that you're looking through is bright red, and then it just everything shows up a little bit funny. Whereas if you have just a touch of it that hits it and dyes the parts that you need, and then the rest is a lot clearer. It helps to see what you have, because sometimes it even the dyes can be like a little harsh and mess with some of the things that you're seeing, kind of wear off some ornamentation on a spore or something like that. Even on certain kinds of cystidia or whatever that have uh, some kind of little crust on the ends or whatever different things, it can wear away at that too. So that's why I try to keep that to a minimum. And then after you have them, it's off to the microscope, where this is how mine's set up with my phone on there, right down the down tube. And then it's
You must have some kind of a mount that holds it there.
yeah, it's just a little thing. It's actually um if you go on like Amazon or something, they're listed around the telescope parts who that also have a very similar size to microscope parts. And it's the same thing. You're looking through a little lens and, hopefully finding a good focus for your phone. But this is usually it's I get in, I find an edge, focus in and out, keep trying to move around I don't know, with the coastline of your little pieces, because that's where, anything interesting is going to be. And just kind of keep scrolling that uh, focal point in and out, especially on, these are pretty big chunks. So that focal point will be further and there will be more depth to those pieces that you're trying to look in and out of. And even, and like the spores that I put on the slide and got the spore drop from, usually don't need them for more than color. Because when you put down little pieces, as you could tell, there's spores everywhere. So you really don't need to gather them up so much usually. But, I like to keep them around just in case there was something interesting. You could just throw another slide right over that, tape it up, and you could hold on to those spores for a long time. But even you could tell the spores that are kicked out of it, of the pieces, are in a different focal plane than the rest of the sample. So you'll find that spot where they're either on the underside of the slip or the side of the slide, and they're sticking right to that side usually. So that's why the focal point is so different. And that's just the rest of it is, I try to keep moving, because I have a couple of pieces, so I can kind of just go quick. And if I don't see anything that's, particularly interesting, it's uh, especially if you have more than one of these type of things that you're doing at a time. uh, it's the difference between, you know, a half hour on one thing or hours doing other, which is why I usually only allow myself to bring a couple home at a time, because otherwise I won't do any of this. They'll get there and then it becomes where am I going to find the time to get set up and make the little slides and do this stuff and scroll all through this, take pictures.
Was that just a cystidium that that you focused on there a minute
Uh, it,
ago?
it could have been. I really didn't find too much on this. Uh, Sometimes, like this part here, I broke it up. Like I pulled the slide out and I tapped on it and moved it around and kind of re-broke stuff up to try to loosen up any material from the big chunks. So it would so then you can get these like smaller clusters of things. Like there's a bisidia right there in the middle left that I'm trying to focus in and out on. and you could see the sterigmata at the top of it. And I'll just, keep going. And if there's something that I see, I'll snap a photo. I mean, I'm not usually running video through it. This is a bit of a first. You could even see the currents from the air trying to exchange. So it's causing currents under there that are dragging the spores and all the material around. So a lot of times stuff is moving too, which isn't always a bad thing as far as I can tell. Sometimes with spores that you're looking for certain shapes, sometimes you could wait for them to roll a little bit. And then sometimes it's nice to just throw in the dark field like that and watch all the little stuff kind of twinkle like stars. And that is pretty much it. That's what we do with them.
That was interesting to see.
Yeah, thank you, Chris.
Do you have an idea about what kind of mushroom that was?
No, not yet. You got any ideas?
I think it's Tubaria. The winter, early spring genus. The spores looked like the right shape. Usually the spores are going to be under 10 microns for Tubaria. The spore print color was right, yellowish, like light brown yellowish.
Yeah, that was a good color that came up. I always go over by the window and try to get some good natural light.
Yeah, Tubaria. Also, Flammulaster have yellowish spore print also, but these don't look like that. Didn't look like Flammulaster. I would guess Tubaria, and I mean a lot of times Tubaria. There's who knows how many species. It's kind of an interesting genus because it doesn't get studied very often. So a lot of times you'll just see Tubaria furfuracea in field guides or online observations or whatever. But I think there's probably a number of species that are lumped into that concept.
It was there in the grass, but I mean about 5 feet away from there, uh, there's a very unfortunate dying maple tree.
Yeah, Tubaria grows on woody debris, but sometimes buried woody debris, or sometimes if there's like wood chips spread, I think the nutrients from the wood chips might leach into a nearby lawn, and then you'll see them fruiting in the grass. So I think that's what that is, but I'm not with real high confidence. And that one thing that looked like a cystidium, that looked like maybe a Tubaria Chylocystidium.
Oh,
That was a
thank you.
nice presentation, Chris.
Thank you.
Can you talk about that dark field bit at the end there? Is that just a filter you slide in there?
Some microscopes even come with that. What's happening there is the light from underneath, instead of going through a lens, there's a little ocular piece there
Condenser?
that you can, like a little diaphragm that you can adjust the light with. So when you're
Okay.
doing
Oh.
when you're doing dark field, there's actually there's a dot, like a piece of material that is now in between the light source and your slide. So the light is actually going around and bouncing around that piece. So that's why the stuff is lit up on a black background.
Okay, I think I understand that. Yeah, okay. Sort of oblique lighting.
Yeah, exactly. Which is there's a lot of different stuff with that. To get things in focus for my setup isn't too simple because I have to actually change. I have a different piece that I have to take out the lens underneath and actually put in this other lens. So it's not on some microscopes, they actually just have like you can scroll through different lighting and different little filters and stuff. And some of them have a little dark field setup built right into them.
Okay.
Does I guess we can do some member finds. I know Dave sent some stuff in. And if anybody has anything from their machine, just put it in the chat. Would you like to go, Dave?
Okay. Yeah, found a few things. It got warm and there was rain, so I checked my early morel spot, but there were no morels. The trees are not even budding yet, so there's no morels out yet, but there were a few other things. So I think these might be Tubaria. That's what I labeled it as. I'm not sure though. Tubaria mushrooms can look a lot different. For all I know, these are Tubaria furfuracea, but these thick stalks are kind of interesting for Tubaria, but I think that's what these are. So I did a little bit of micro on these. Now, what I do to look at spores is I put a cap right onto a mushroom slide, or right onto a microscope slide, and collect some spores right on the slide. And I was pretty sure these were Tubaria. It was a very light spore print, and so it wasn't highly pigmented, and it wasn't very thick either. But I could see it was a little bit pigmented, which reminds me of Tubaria, kind of a not highly pigmented yellowish light brown kind of spore print. And notice these spores they're pretty much ellipsoid. They're not showing a very prominent apiculus. So, Chris, these look like your spores, that you just showed. And I think these are Chylocystidia. I just did a smash mount and I did find these things along one edge, one part of the perimeter of the smash mount. And this is pretty much what Tubaria Chylocystidia are supposed to look like. They're pretty variable in shape. A lot of times they're cylindric. Sometimes they're a little inflated toward the bottom or toward the top, and sometimes they're described as flexuous, which means they're kind of like curvy. So I think that's what this is. It was in forest litter, just amongst leaf litter and some twigs and things in a hardwood forest. So I think that's what these are. I dried these out. I'll send them in for sequencing. Maybe we'll see. There's a lot. I have a lot of stuff, that I could send. I'm gonna have to like pick and choose eventually what I send. If these are fairly interesting though, because I'm not sure what they are, so I'm calling it Tubaria for the time being.
Nice. Oh wow.
So I went back today. So these were observed on Saturday. And it was a warm day. There was rain the next day. There had been rain before this, a fair amount of rain in this spot. And these were surrounding a pretty much healthy looking tulip poplar tree. And for the last 5 or 6 years, I've been finding these Coprinellus mushrooms around this same tulip poplar tree. And it's also a spot where I usually find a few black morels, but it's too early for those. Maybe in a week, we're going to need to have another warm up. It's going to get really cold now, so that morels will be on hold. But these are Coprinellus section Mycacei or Mycacii. I'm not sure how to say that. So these are called mica caps. There's more than one species, but I think these might be Coprinellus truncorum. And the reason why I think that is because, well, I went back today to this same spot, still no morels, but there were a few more of these, and I wanted to bring home one of these to look at because, as you see on these photos, it looks like the stipe is smooth. I don't see a whole lot going on. It's not real hairy. In fact, there's a next photo, I think, shows the stipe surface through a loop. And I'm not seeing any hairs on the stipe. So what I did not do when I collected these on Saturday was I did not examine the stipe Stephanopellis through my microscope to see if there were any caulocystidia. But today I went back and got some more and I looked for caulocystidia. Anyway, these are the spores, variably shaped depending upon how they profile. Sometimes ellipsoid, sometimes sometimes mitriform. Some of these are mitriform. Mitriform is shaped like a bishop's mitre, the hat that a bishop wears. And some of them are mitriform. This one right here, right in the middle, that's pretty much mitriform. Now the other ones are more or less just irregularly shaped. But notice how they're all truncate. On one end, there's like a really large germ pore, and it's like the spore is cut off at that germ pore. So that's what it means for the spore to be truncate. So these spores were observed from a spore drop. One reason why I like to observe spores from a spore drop is there's not much clutter, and all the spores, you tend to be able to get the spores squashed down into more or less one thin plane. So a plane actually has no thickness, but when I say one thin plane, I mean a really, really thin mount so that it's almost a plane. And this picture right here is the stipe, the Stephanopellis. And the reason why this is significant is there's no cystidia coming off the stipe surface. So caulocystidia are projections, cells that would be projecting off the stipe surface, and this is not showing any. And this was one photo that I took, but I looked at several areas on two different mounts from two different mushrooms of this stipe surface, and I couldn't find any caulocystidia. So I'm going to say these are probably Coprinellus truncorum. Now, what the heck are these things? I think these might be Pleurocystidia. I'm not sure. I mean, this would be weird looking Pleurocystidia, but I think that might be what they are. Now, this I found in the stipe mount. So why would they be on the stipe? Well, presumably maybe they fell off the gills and stuck to the stipe. So these are not spores. These are way too big to be spores for this species. So I think I just added these photos like a minute before we started today, and I haven't really had a lot of time to think about what these are, but I think these are Pleurocystidia.
Dave, that's pretty interesting. I'm wondering because when I see those kind of, I call them kind of balloon shape, but you can only
Yeah.
see the top of those balloons when they're actually, you're looking at like a palisade or something like that. So
Yeah, when they're embedded in the gill trauma.
right. So I'm wondering if that's what I'm used to seeing and calling.
Yeah, I don't know. A lot of times they'll call these kind of things terminal cells when you see them on the gills.
Yeah.
But I did do a little bit of online searching before we started today. And there was one photo that I found online that showed what they were calling Pleurocystidia of a different Coprinellus species. It wasn't one of the Mycological species, but it was not all that different from this. So I think that's what might be what these are. But and there's one that's just sort of wedged in with the hyphae making up the stipe. So there's no what do you call them? What are those connector things?
Little clamps.
Clamps. Yeah, there's no clamps on these either. Yeah, you can see that pretty clearly. So anyway, I'm not sure, but I think these are. I'm going to say they're probably Coprinellus truncorum. I dried some out as usual. I have a sample. So every year I find them in the same place, and it's interesting because this tree is not dead. Supposedly, these Coprinellus mushrooms grow on dead wood, dead roots, stumps, and so forth. A lot of times you'll see them on stumps or on the roots connected to a stump. But this tree seems to be alive. So,
Well, the worms are clearly happy. They're eating something. So, maybe it's just all the forest detritus.
well, I'm sorry, worms?
Yeah, you could see all the little bits around where they're coming out. You could see all the little
Castings,
that's
worm
all.
castings.
Oh,
That's all worm dirt.
oh, okay. I didn't even notice that. Huh. I'll have to have a closer look at that myself later. Oh, thanks. That's those. They call them mica caps. Usually they'll have little shiny grains on the caps that look like mica deposits. These are probably got washed off by rain. Last week we had rain almost every night. And the cap surfaces are fairly long striate. So they have those radial grooves. But that's us. Yep, that's Coprinellus. It's considered to be an inky cap species, but they don't particularly deliquesce all that much unless it's really, really damp and moist outside. Usually they'll just dry up. Now this is interesting here because what I find interesting here is because is Hunter here today?
No, she said something last week that she wouldn't be here, but the
because
picture from the email.
The picture in the email looks like this, doesn't it?
It does.
Yeah, and I thought for a minute that's what hers were. But hers, well, it would have been good if she took a photo of the undersides, but one of the fruit bodies in Hunter's photo, which was identified, and I supported the identification as well. I agreed with it on INET. Dacropinax elegans, I think it was. It was a Dicropanax species. I think Dicropanax elegans. It's like a plasticky jelly mushroom sort of thing. It's not real jelly like though. It's more like plastic. But notice that I know that mine are not Dicropanax, even though they look like hunters. And the reason why I know mine are not Dicropanax is because these are gyrocoid. They have gills. So this is actually Tectella patellaris, and the common name for Tectella patellaris is veiled panis. So these used to be in panis a long time ago. But you might be wondering, what's up with the veil? I don't see a veil. These things persist for a long time after they fruit, and they tend to fruit in cold weather and hang around most of the winter. You can barely see a little bit of velar remnant on the one. If you go back to that photo, the one in the middle, see there's like a little bit of white clinging to the margin of the cap. That's the remnants of the veil. So this thing had a veil over the gills when it first fruited. These things had, and then they expand and the veil tears apart and erodes away. There's a little tiny bit left though. But I know this is Tectella patellaris. I found this species before, and you usually find it in winter or early spring. But from this side,
one in the upper right, you can see the partial veil, or I don't know what you call that veil in these things, but
yeah, it's a partial veil, I guess. Yeah, it's a partial veil. Oh, okay. You can see better in that one in the upper right. Okay, but viewed from this angle, though, they look like Dicropanax. It looks like Hunter's photo quite a bit. So I found that kind of interesting when I saw Hunter's photo. My first thought was, oh, Hunter found Tectella, and then I saw the identification on INAT. But I looked closely at her photo. There was a little piece of cap where the underside was kind of upturned and sort of bent around, so you could see a little bit of the underside, and it didn't have gills, so it wasn't this.
Yeah, I took a second look at that one also and thought the same thing.
Yeah, so that the underside apparently was smooth, like it should be for Dicropanax. I got some spores to drop out of it. These are white spored, by the way, which is you would expect from the gill color it'd be brown, but these are white spored. You pretty much have to stain the spores to see them at all. They're small, shaped like little sausages. They're very small. These are very small. They're probably only like 1.5 microns wide. Once again, I got these spores I found from a drop. So I like to look at spores from a spore drop because then there's not a lot of clutter in the way. Now, there is clutter in this photo, but that's just because there's scratches on my microscope. So my microscope's not in the greatest condition. But it works. It's easy to use, and I can work very quickly with it and get a lot of photos. And look at a fair number of mushrooms in maybe an hour or two.
Yeah, that working quickly bit. That's a big part of it.
Oh, it's a big plus. That's one reason why I'm reluctant to move away from this scope. This scope is old and beat up, and it's simple, but it's easy to use. I can work very quickly with it.
It first appeared on December 6, 1877. What I noticed or found interesting about that observation, Dave, was the central stipe on that.
It's not quite it's kind of like a stipe.
Kind of, yeah.
It's kind of like this cap is just sort of elongated laterally. And it does look like a stipe though, doesn't it? I guess you would call that a stipe. It's sort of just like the cap though, it's just sort of stretched out where it attaches to the substrate.
Yeah,
And
but you look at it from the underside and you can see that right,
yeah, you can see where
it's
the gills all terminate.
all, radiate from around that. So
Yeah.
it's very interesting.
Yeah, it's an interesting little species. You find it in cold weather usually. Grows on hardwood sticks. So I didn't know what this was. I put it on INAT and Mushroom Observer, and John Plischke came on and said and identified it as Trichaptum abietinum. Now this is growing on hemlock. This was a big old hemlock trunk, a tree that fell over and was probably they cut through it to get it off the Falls Trail at Ricketts Glen. And I did not recognize this as Trichaptum for two reasons. And John's probably right. I agreed with him. And somebody else came on too, Ariel Bokonski or something I think she's from Minnesota. She does a lot of identifying on INET. The things that kind of support Trichaptum are this purplish underside. You can see it's got kind of a purplish tint to it when you look at it from afar. The two things that sort of made me scratch my head about this being Trichaptum are those things are coming up in the next photos. I didn't get any spores out of these, by the way. Nothing fell. These caps are really hairy. They're very hairy. I'm not used to Trichaptum being this hairy. Like these hairs are pretty big. I think I have another photo as well where I show. Yeah, there's even a little closer. You can see this probably through the loop. You can see it's pretty densely hairy. Now, if you read the description of Trichaptum abietinum, though, the descriptions do say that the caps are hairy. So, but then if we move on again, what I'm usually used to seeing in Trichaptum are the elongated, lacerated tube walls. And there's one more photo here that'll show a close-up of what we're seeing here. So what we're seeing here is just sort of this dietylate pores, that not quite dietylate. They're not really all that long, but they're they have twists and turns in them, and the walls are not elongated at all. But I'm saying I guess it's just because these are fruited. They either recently fruited or they fruited right before it got cold, and they really haven't changed very much. So this is what I imagine, if this is correct, which I'm not saying it isn't correct, it probably is, Trichaptum abietinum. But
Dave,
the pore surface.
how big were the caps that we're looking at in that little cluster there? Is it small or
maybe 2 inch? The biggest ones would be like 2 inches. I think I forgot to put a penny.
That's even so I think I know Trichaptum abietinum pretty well. It loves the pitch pine here in South Jersey. This is pretty meaty also.
Yeah, they're kind of thick. Yeah. But they're young, and I imagine that species, the fruit bodies actually thin out and spread out a little bit as they mature.
I think that's likely true. And oftentimes when I find what I call Trichaptum abietinum, and what's her name, Ariel, and others come in and they agree. I think it's kind of a well worked out thing. But I can recognize it right away, and this looks atypical, and
it is not typical. This is I posted it as polyporalis or whatever, because I was not taking any stand on what I thought it was. Quite frankly, I didn't know what it was. And John and Ariel came on and both said Trichaptum abietinum. And so when I thought about that, I thought, I cannot disagree with that. It's the right substrate. It's conifer wood.
Yeah, the way that I find Trichaptum abietinum, it's okay. You found a Trichaptum, a thing that looks like a Trichaptum.
Yeah,
it's on conifer.
Yeah,
Okay.
that's pretty much the way I identify this. These just didn't look like Trichaptum to me.
Exactly. That's what I'm saying. Doesn't quite it doesn't quite fit the bill. I kind of agree with your, you know.
Yeah, I have it on Mushroom Observer as promising. I'm not taking a strong stand on it being this. I preserved it. I've got a collection preserved. And this would be a good one.
Yeah, I
It's
agree.
funny. Like I mean, I have all this stuff I'd like to submit for sequencing, but I'm not going to be able to submit everything. I don't know how much stuff I have from 2025 alone. Probably 400 or 500 samples. So I'm not going to be able to submit everything. But
That's that's a lot.
it's a lot of stuff. But but the growth pattern is right too. I mean, this is sort of what Trichaptum does. It forms these shelves. They're not big. Trichaptum polypores are not big. These are not particularly thick either. They're just kind of really hairy, which that kind of threw me off a little bit. I thought it was an interesting one though.
I think it is. It should be moved up at least in the top 100 of
Ah,
your potential.
I'll see what I can do, but I'd like to find out. I'd like to find out about this one.
the top 100, man. Is that it?
Yeah, I think I got.
That was it.
I think I've got two more. I think I sent 5.
Yeah.
Yeah, so that was the third one, I think, wasn't it?
No, look at that.
Oh yeah, it's Steccherinum. So now Steccherinum ochraceum is generally it's supposed to be this cortisioid thing. This well, maybe not cortisioid, but at least resupinate. It's just this thing that's plastered on wood, and it's got spines for the hymenium. Well, these are little caps. And in the past, I found what I think are little caps that are Steccherinum ochraceum. And when I first found these, I was puzzled by these as well. It wasn't until I got them home and started looking closely through my loop and I saw the truly hydnoid hymenium, as you'll see in one of the upcoming photos.
Looks like there's a stem on there, right?
Yeah, there's a little stem on that one.
Have you considered Bordeauxii, Steccherinum Bordeauxii?
Bordeauxii. Is that hydnoid though?
Yeah, I'll have to look it up, but yes.
Let me write that down because I don't know that one. Bordeauxii. Okay.
And you think that's the little stipitateness, and it's not because of the moss or whatever. It's trying to get out past the moss or something.
They were growing on top of a beech log. And I think there was some moss on the beech log. I think there was moss on the log. Yeah, there it is. There's moss on the log. You can see it. But if you go to one of the subsequent photos, maybe like there we go. I mean, this is truly hydnoid. This is not like Trichaptum, where it's just tube walls that lacerate. These are actual spines. And I think is there one more photo of?
Yes.
Maybe not.
I just looked it up. Steccherinum Bordeauxii. Most of the Steccherinum are truly hydnoid with similar looking teeth, and these teeth look like Steccherinum ochraceum.
Now, these spores that I got really fit Steccherinum ochraceum. They are truly ellipsoid. They're not allantoid. They're not like little sausages. They are truly ellipsoid and very small. These lengths are not much past 4. Maybe the long ones are 5 microns long. but they are ellipsoid. They're not allantoid. They're not like little sausages. They're ellipsoid. So that does fit Steccherinum ochraceum, but maybe it also fits Steccherinum Bordeauxii. I'm going to have to check that out later. Thanks for the tip. It's give me something to do later.
Hmm.
I think I have one. I think there's one more. Oh, and by the way, today I found some nice Xeromphalina. And I've had Xeromphalina sequenced a few times, and more often than not, it's coming back as Xeromphalina enigmatica. So the enigmatic one, the one that's not Kaufmannii or Campanella. Was that it? Were there
I
5 of them?
I think that was it.
Oh, okay. I thought I only counted to 4. Oh, let's see. There was the Tubaria, the Coprinellus, the Steccherinum, the yeah,
Tubaria.
that's it. That's 5. Yeah, thanks. Sorry, just lost count. That's it.
Nice. Thanks, Dave.
Sure. Yeah, I have time. I'm retired. I've got spots I can go to in the woods. They're not very far from my house, so I try to get out a lot.
Nice. Emily says she's got one to share. You want to share it from yours?
Sure, give me just a second here. Pull this up. So my magical fire pit has come up with something else. Hang on here.
Not a morel.
I don't think so. All right, can you see my screen?
Ooh.
Yeah.
All right. So when I first looked in there, I was looking for the morel that I'd seen a couple years ago. And I found this little guy, and at first I was like, oh, it's part of a morel. And then no, no, I don't think it is. I haven't done any research yet, but I'm pretty sure it's a cup fungi. He had a little friend munching away.
Nice.
And
Oh, it's a snail.
yeah, one snail and one little cup fungus that I haven't done any research on yet. But if somebody could point me in a direction, I'll start doing some research.
I mean, Helvella, but I don't know. I don't know these cup fungi all that well because it's pretty big, right? Well, that
Yeah,
snail's small.
yeah, the snail's really small. I would say it's maybe the size of a quarter.
Yeah, yeah. Pezizales is not a bad idea, but I don't know if they're. Yeah, I don't know them well enough. I don't know the big cup fungi well.
Yeah, neither do I.
I mean, this time of year, the first ones that I start finding are those I mean, what I think are varia or whatever.
I think this is one of these stipate Helvella things. There are these Helvellas that really are very cup shaped, but they have this like fluted central stipe.
Yeah,
And
that's what I was looking at too, Dave. Even in this
yeah,
photo, if we can zoom in a little bit, it looks like we have that ribbing or fluting.
yeah, it
Yeah.
looks like one of these stipate Helvella. There's like 2 or 3 species that look like this. I don't find them in the spring though. I usually find these in the summer. But I think let me try to get a name here. I'll just grab a name off of Mushroom Observer or a Mushroom Expert or something. There's a few of
Yeah.
them.
The interesting thing is the dark hymenium with everything else being white.
Mm-hmm.
I think is pretty interesting. Or is that just snail goop?
I mean, he is a black snail.
Because I guess it could be like a shadow or something too.
No, I definitely think it's darker. It's a little dark outside now. I could go out and check.
What about compressa, Helvella compressa?
Compressa?
It's my chair. My chair.
Are they usually like that too? These Helvellas, like cup on top, stipe on the bottom?
Yeah,
Oh, they seem to have a pretty long stem though.
yeah, but maybe that one is longer if it went down through those needles, you know?
No, I don't think this is compressa. Compressa has a long, thin stem, like with just. No, I haven't found the name yet, but there's like 2 or 3 things that look like this, and I'm gonna find. You know what? I have an observation from last year on Mushroom Observer, and I'm going to be able to track that down by doing an advanced search right now because I know where it was found. That's just going to take me a second.
Is there any significance of the fire pit? I was not expecting to find yet another mushroom in my fire pit this year.
The Ascos love a lot of Ascos like burn sites and fire
Yeah,
pits.
Yeah.
there very well could be.
Well, I'm definitely going to keep checking it. Never know what I'm going to find next.
Oh, absolutely.
And was this you at least upturned it? Was it very brittle, or was there a little give to it? Like did it
No,
fall?
it wasn't brittle. No, it was pretty solid.
Yeah, I think that supports Helvella too. A lot of the true Pezizales genus, they're somewhat brittle.
Yeah, I think Helvella was the only thing that INAT had suggested, but I wanted to do more research before going with it.
I'm going to put something in the chat. It's not the same thing as what you have here, Emily, but it shows like that we're on the right track, sort of, I would say. So I'm just going to put this MO observation right into the chat. And who is hosting? Is that Lila? Or Chris? Is Chris host? Maybe the host can just click on the link that I put in, and you'll see that it's what I've posted. It's not this, but it's similar to this. There are a few differences that I can see right off, but it's sort of in the ballpark with this thing.
Yeah, I just clicked on that link, David. It's not dissimilar. Obviously, it's not as veined on the outside of the cup.
Yeah, this one, the Emily's doesn't have the veins going on the underside of the cap.
No.
Yeah, so it's different.
But again, there's obvious fluting in the stipe.
Yeah,
there's no question that that's a thick stipe, and I think you got all of it there. I don't think you missed any of it.
If anything, it might be a little bit attached to this
Yeah.
little bit of bark down here.
Yeah, you've got the whole thing because you can even see the substrate is still attached.
And I'd call that conifer.
Mm-hmm.
Now with
conifer.
With this thing, you can turn it upside down with the concave part, the up side of the cap. You can just turn that upside down. A lot of times, if you're lucky, you get some spores to fall out, and then you can scope the spores. Helvella spores are really big, and they usually have a great big central gutule. So I think almost all Helvella spores look like that. Like they're wide, they're big and they're wide, most of them, and they have a great big central oil drop. So it looks like a dilated eyeball or something. But if you can't find spores, if you can't get them to drop, which a lot of times they won't drop on an asco because the ascos just sort of fire their spores off every once in a while, and if you pick it when it's not going to fire spores, you just won't get any. But you can scrape a little bit of that gray inner surface,
Hmm.
and mount that on a slide. It's not hard to mount that stuff because it's soft, so you can squish it pretty easily. And then you'll see the asci, and you'll see a few spores floating around. I would suggest using a stain. All of these ascos have white spores, and they might be hard to see in water. I'm not sure. I always use a stain with stuff like this.
All right. Yeah, I'll see what I can do. Thanks.
Sure.
Thank you. Keep checking the fire pit.
Yeah.
Paul, did you want to share from your machine?
Yeah, I think, doesn't Jamie have some? I think she's ahead of me.
Oh, go ahead, Jamie, if you like.
Okay, hi. I'm gonna share my screen. Let me see.
Oh, Jamie, while you're getting started, I just want to mention, I'm not sure if you stayed in that one session a few weeks ago, but it was after the session where I had said that I thought I found Octospora.
Yeah.
They were not Octospora. And in fact, one of the crucial features was that and Lila had both pointed out was my observation was made um,
Yeah, it was on
habitat that had
soil in a flower pot, right?
no moss at
Yeah.
all. Yeah, so it's a different. I forget the genus name. It's something I had never heard of. Nicholas van Vuren from Holland identified it for me on ASCO France. Anyway, if you're really interested, um, I'll put my email in the chat. If you're really interested, I'll send you the observations to look at.
Okay, sure. Um, I found, so I wasn't here, I think, the last week or two. Um, but it's been warming up, so I've been finding some cool stuff. Um, this one, I feel like I normally, when I find these, they're older and they start to look kind of decrepit. But this one that I found, it was like pretty fresh, and it had this really interesting ring on the underside. Um, so that was pretty neat. I don't know. Can I see the chat when I'm
Hmm.
um but yeah, this one was pretty cool. I found it while I was doing a stream assessment. So this tree, you can kind of see it gets like sometimes when the water floods it, that part is below the embankment a little bit. So it's coming from a spot that can be underwater at some points, which I thought was interesting. But yeah, this one I'm pretty confident on the the identification, but still
Are you certain
pretty
it was
cool.
growing on hardwood?
It was like coming out of the roots here, so
Yeah, the reason why I ask is there's a variety of curtisii that grows on pine roots.
oh, interesting.
Really?
Yeah, I forget the name of the variety, but it's the Ganoderma curtisii variety something, grows on pine roots, which I have found in not in the New Jersey pine barrens, but in Cape Cod I found it in a pine barren setting.
Hmm.
That's
Yeah, because
interesting. Yeah, I
yeah,
don't
Jamie, I would call this curtisii in a heartbeat. Wasn't
It-
wasn't aware that if there was some pine in the area, um,
yeah, they
which
look the same.
yeah,
The two versions of curtisii look the same.
yeah, I think it was a deciduous tree, maybe.
It seems to be the more common setting for curtis. The other thing seems to be uncommon. I only saw it the one time, but I don't visit habitat like that very often though.
Okay, and then this one I found, when was this? This was on April 3rd. Um, and it was pretty cool. It had so many fungus beetles and flies on it. It was wonderful. It was like I saw the cool fungus and then I saw a billion other cool things. Um, but I thought this was probably some sort of oyster, and when I put it on INAT, it said oyster mushroom. So I don't know what the consensus is on it, but it was
To get a spore
pretty
print?
big, pretty soft. I did not.
Yeah, if you get a
Um,
spore print,
but
usually the colors will vary between some of the different ones.
Yeah, the cold weather oyster before actual spring arrives, like in mid-May, is usually Pleurotus ostreatus. And they are usually a little darker on the cap, like brownish or grayish. So I I think that's probably what these are. So in the spring, when once it warms up, and it's like we're well into morel season and even getting past morel season, you'll find a white one that grows on poplar, true poplar, not tulip poplar, like aspen, and that would be Pleurotus populinus, and that one tends to grow in later in the spring.
okay, very cool. And then um, this was in the same spot that I found um the ratio. Um, and it's just a little canker, but it was pretty bright orange, so I'm not sure like what the life cycle is like on them, like if they look orange all the time or if this one is just orange because it's like getting ready to reproduce or something, but it was on also like a piece of root that was sticking out of the embankment.
I feel like the more damp it is, the more orange they seem.
Okay, that's cool. Because I've seen them on like tree branches, like around eye level or higher before, but this one is it was kind of like below ground level because of the embankment. Um, but yeah, this is also just the what INAT suggested. So if anyone disagrees with INAT, let me know.
Well, then I just stepped away for a second, so I came back to just this photo. We saw the chestnut blight presentation last week. I suspect there's more that meets the eye than just that one, and then this Endothia gyrosa. Um, and I suspect they're very host specific, but yeah, everyone just kind of calls these the orange hobnail canker, but
Okay.
I suspect there's more that meets the eye there.
Yeah, I got one of those
Or,
I was gonna show.
oh, let me see. Did you put it in the chat? Is that what just popped up?
No.
I just put into the chat a Helvella that looks like it might be a match for Emily's Helvella. Helvella solitaria. So I found it on Mushroom Expert.
I've got a couple fun slime molds that I found. Um, so this one, INAT said, um, oh, this one, INAT told me genus Metatrichia. It looks like someone disagrees. Um, but it's got that like round
Oh yeah, that's
appearance.
Nice.
And it was really cool because I flipped the log and I saw like a bunch of little clusters of them next to each other, and like some of them were still closed like this. Then some of them had like just started to spore, and then some of them were just like totally, um, you know, crazy.
yeah. I know what this is. It used to be Trichia proper. That's probably why Lee just kind of downgraded it to Trichiaceae. Um, gosh, Bulliginia, I think was the epithet that it used to be, or there's more than that species. They used to just all call them one species. I'll look it up. I'll find it. But just of note, if you want to know that you have something in Trichiales at the very least, colorful capillicium, so that cotton candy bursting out,
Yeah.
that puts you in order Trichiales. So that's a good way to kind of, if it's brightly colored and not, just dull black or gray, that's a good place to start.
Yeah, and I've gotten pretty familiar with the wasp's nest slime mold because I've been finding that one a lot. So a lot of times when I see other things, I'm like, oh, that kind of looks similar to that. So it's probably in like the same family or the same order at least.
And good so yeah, you're familiar with the wasp nest. A good note when you're looking at slime molds is there a stipe? What's that stipe look like? And how many of those individual little fruiting bodies are at the end of that stipe? So if you go to that other photo where yeah, here, I bet you there's little to no stipe. And if there is a stipe, it's very inconspicuous.
Yeah,
Where,
they were like super squished onto the surface.
yep. Whereas, as you would know, with Metatrichia vesparia, there's obviously a stipe. I mean, these
Yeah.
things are small, but you can make that out pretty clearly.
Yeah, it looks more like the one that's on this
So
this
how big are
little
these little
genus cover.
the little round beads here? Like a one of those little round beads. What is that? Like a centimeter, half a centimeter, couple millimeters?
Sorry, are you asking like how big this was?
Yeah.
Um, I have a picture. I might have not included the one that had the little tiny isopod on it. Um, but I mean it's pretty minuscule. Um, I would say that this whole patch is probably around the size of my pinky nail.
Okay.
So I took this picture with my phone with my jeweler's loop. So it's pretty small. But it was hard to miss because it was so brightly colored.
Brandon, are these the things that have the weird the weird strands in the capillicium that are like braided?
Yeah, they will have banded or spiked capillicium, so there'll be these little spikes coming off of them. I'm trying to find my observation, but uh, observations, but I'm having a tough time with iNet right now. It's not responding.
Okay. Well, I'm sure that you'll probably chime in at some point on it because I've seen you IDing some of my observations.
Yeah,
Um,
where did you find that?
this one was at Clayton Park, which I've been going to recently. Ton of spring ephemerals there. And Alan Crawford, I don't know if any of you know him, but he's like a naturalist and an illustrator. And he said that he called it a mycological wonderland. So he said, go back there on like a warm, rainy night in May and you'll find some crazy stuff there.
Yeah, I go to that park all the time. I love that park.
Yeah.
There's
I've been
either a
to
lot or
before.
there's nothing. It's like
Sorry?
there's either a lot of stuff to find or there's literally nothing.
Yeah, I mean, I lately it's been really cool because all of the spring ephemerals came up, so I got to see some really cool species for the first time, like out in the wild, like round-lobe hepatica. But yeah, this was found flipping a log. Um, I found some wasp's nest slime mold there, a bunch of the like classic polypores that you see. Um, and some of the fungus beetles as well with it. So it seems like a pretty healthy ecosystem there.
Yeah, I
This
think
one is one that I found today, and I was like, "Oh my gosh, it's so crazy looking." Um, I was very excited when I saw it. Um, like this looks like boba, or like the eye stalks of like a little snail or something.
That's cool.
But um, it's like I can't tell if this is like a separate crust fungus that like these are just kind of like growing on top of or like hijacking. These ones they look like they're a bit more dry. Um, or if this is like, the main body of it, and then these are just like the fruiting structures. I don't really know what's going on. Um, but
And while I share a little bit of your, I don't know what's going on there either. Um, Comatrichia is probably a pretty good idea here. But so with these type of fruiting bodies for slime molds, um, you can analyze what is called a pseudothallus, but that pseudothallus is never this kind of membranous. So I think there's something else going on here. And I think you can also see those fruiting bodies in one of these photos coming both off of whatever that
oh
membranous
yeah,
material is,
like
as
right.
well as not.
But yeah, they're pretty cool. I found these in the grassy marsh at Tuckerton, which is the little spot that I'm doing a field guide for. So I was like, yay, another slime mold that I can add to the field guide. Um, because I'm trying to like get it as representative of all the different taxa as possible. So the more weird stuff, the better. But yeah,
Agreed.
that's the
And
genus that was suggested by INET. Um,
just so you know, if you're interested in these, if you find this in this state, if you take this photo, whatever time you took it, it says 122, uh, and then go, collect or identify in other places. A couple hours later, you might have a little more information here. Some of these might start opening up. It happens pretty quickly.
yeah, I'll probably be back on Thursday, and I know what log it's under, so I'll go take another peek at it. Um, I would be very curious to see if they, you know, like explode with spores or something. But yeah, that was just super exciting find earlier today.
And
Um,
just because I'm on slide mold brain right now, those wouldn't explode with capillicium like the last one we saw, which I found the name for, Oleaginema. I'll put some in the chat. This that peridium just kind of cracks open, um, and the capillicium kind of stays in place, and the spores are blown through the capillicium. Sometimes some
oh,
capillicium
okay.
will you know make it farther than that peridium was, but these won't throw that capillicium. And when I say capillicium, I mean cotton candy, right?
Yeah. Okay, that's cool. And then literally like two logs down on the trail, there's this um decomposing, I think it's a Atlantic, and it's got this like little pocket in the back that's like super of water and it's damp, and then it keeps this like moss damp. And I saw like little tiny like younger versions of this a couple months ago when it was a bit warmer, I think in like January for a little bit, but they finally um a couple weeks ago actually like got big and looked like this, which was very cool. Um, INET suggested the family Sarcosomataceae.
Yeah,
But I
that
don't know.
seems
All I know
like
is that it just
that
looks.
seems likely. Lila, you know this one, right? Is this? I always want to say Bulgaria.
There's this thing that looks like Bulgaria,
Yeah,
and I think it's this. Um,
yeah, I
I
don't think
forget the
this is the
name.
Bulgaria iniquens. This is probably some sort of sarcosoma.
Hmm.
But it was interesting. I tried to see if there was any sort of like visible um like spores or like pores or gills or anything. It just looked kind of like smooth on the inside. You can see um like it looked a similar texture on the like inside of the cup as the outside. Um so I'm not
Well, if
really
it's
sure.
if it's if it's Sarcosomataceae, or in the family of Sarcosoma, isn't that like kind of liquid, or like a gumdrop consistency that breaks away?
Yeah, I was gonna ask, is that related to that thing that Sue Hopkins shows us every year?
Yes, I think it is.
That's cool.
But yeah, I just went back. I got really busy at work, so I didn't get to walk around the trail a lot. This was March 26th, and then when I was walking around earlier today, I think only like this one was left. Like the rest of them were all um like there was kind of just like some mushy spots, and then I don't know if they got like eaten by something or if they just kind of like disintegrated. Um, but there was not much left. So
I think I got what this is. I just put it in the chat. Pseudoplectonia nigracians. Or Pseudoplectonia nigrella. I just put it in the chat. It's something that I found early in the spring one year. So there's a Mushroom
okay.
Observer link. Chris, could you click on that thing that I just put in the chat? Because I think it's this. It looks the same.
It looks like it has a different like
Is
text outside. Oh, sorry.
was
I think
this
I'm only sharing my one screen. Um,
Yeah, I mean, you can, since you're sharing, you could grab that thing that Dave has and you could share it on the screen.
let me just share my
Hmm. Dave, yeah, good ID there, I think. Or at least it matches very well what you have. Yeah.
I think that's what this is. No, it's not very common. I think I've only found that thing once. It was early too. There's a date on that
March.
mushroom. Yeah, March 17th.
Okay, so
Yeah.
that's only like a weekish difference. Like not even 2 weeks. Um, so similar time of year.
Similar time of year. Yep.
Okay, yeah, that definitely could be it. Yeah, thank you, Dave.
Sure.
Where did you find yours?
It was on the base of a mossy oak tree in hardwood forest with probably some hemlocks here and there.
In Pennsylvania.
In Pennsylvania, right? Yeah, in Pennsylvania.
Not at all coastal.
Yeah, not a coastal
That's why,
thing.
like, I'm always curious, like, what grows here and how different it
Right.
might be from everything else. Like, this area, like, this very log probably got flooded sometimes because we've had it come up to, like, the back step of the building before, and that's definitely higher elevation than where this log is. So it must be at least somewhat salt tolerant. Um,
Let's see if Michael Quebec
Yeah,
has
I
this
was wondering,
species.
Dave, where'd you get that name? That's that's a name, all right. And I was looking at your observation. Rene LaBouffe agrees. So
Yeah, I forget how I tracked that down. I think I thought it was might be Bulgaria, And then I just looked up like things that looked like Bulgaria. I looked for lookalike species because that was my starting point with that. I'm pretty sure. Okay, Mycological has Pseudoplectania nigrella. What do they say about the habitat, growth patterns, solitary, dispersed, gregarious, or in clusters ecology? Among the needles, grasses, or mosses under conifers and on mossy logs. Okay, mine was not really in a coniferous area, but I'm pretty sure it was right. Period. Spring, April to June. That's in Canada though. April in Canada is like March down here. Um,
Yeah, and they both were in like mossy areas because you said yours was in
it was
mossy.
on a mossy base of a tree. Yep, a big oak, if I remember. I forget if it was alive or dead.
Okay, and I've got one last one. Um, this one is not a shelf fungus that I've seen before. Um, or if I have, I don't remember it. It's got like an interesting, it almost felt like velvety on the underside. Um,
Mm.
and it, I don't know how to describe it. It was like very flimsy. Like you could see here, I just kind of like folded it back. It's not firm like a turkey tail or um, even with like a stereum, it's still got like a little bit of um, like rigidness to it. This one was like very soft. It was growing right next to a stream. This was in right off of 72 at one of my stream sites, and this is technically in Greenwood Wildlife Management Area. Um, and I was like further in the woods a little bit. It's on this kind of like fallen log right next to the stream. But yeah, I'm not really sure what it is.
Is there an in situ photo? Just kind of how you came upon it.
Could you say that one more time?
Yeah, is there an in situ photo? Just kind of this thing undisturbed, just how you found it.
Let me see. That's what it
There-
looks like, just like
yeah,
on
there we
the.
go. Wow, that's interesting.
So like it grows like folded and it looks like it would be kind of rigid in that shape, but then it was actually like super soft and you could just kind of like bend it whatever way you want. Like I
It
just folded
doesn't
it like this
have,
and it didn't break off.
and it doesn't have pores or that hymenium is unfamiliar to me.
It's kind of marulioid, isn't it?
Yeah, kinda. Yeah.
What, and it's dark on both sides?
Yeah, but on the upper side especially, it's got this like white cream colored lip.
Mm-hmm.
And that seemed to be pretty characteristic on all of them.
Yeah.
Um, that looks like it's like a younger one that's about to pop out. Um, I'm not sure what kind of tree this was. It, it's like a pine barrens habitat, so there's, um, some oaks, there's cedar. There were quite a few cedars where I was, so this could be a cedar, especially considering it's not like totally decomposed yet. Um,
And I know it doesn't like because of the I don't know. Could it be a beat up auricularia somehow? I mean, like with that kind of texture.
I'm not sure if auricularia is the best idea, but I had a similar thought that it could maybe be infected by something like uh, that hymenium is just very odd to me. It, Morolioid or some kind of jelly makes the most sense here, I think, right?
yeah, I mean, it did definitely seem like it looked like a shelf, but then it felt more like a jelly. Um, but I mean, even with like a jelly fungus though, it's more like smooth and kind of goopy. This one was just like velvety on the underside.
Well, velvety would you know support some sort of infection, right? Secondary, uh, fungus coming in there and parasitizing whatever this is. But
What's what's
even
right
that, I don't.
beyond your hand? What's right beyond your hand? What is that?
Oh, this guy?
What is
Um, I think I put some other observations up. I think it was just as tremites for now. There was a lot of like other shelf fungi around.
maybe some of these are infected and some aren't. If you have something similar down the log in shape and size.
I've never seen a Trametes infected with anything that produces this critter.
No, it's weird.
This,
I'm kind of stumped by this. I don't know if anyone else has any ideas.
No
Yeah,
clue on this one.
I've
yeah.
seen I've seen some trimmings that are like infected by stuff before, and it usually just looks more like a little like fuzz that's growing on the underside. Like I personally haven't seen something that like changed the whole color. Um,
Did you save any of these? These are very interesting.
I didn't, but I know where it is, and I could go look for it again. Um,
Yeah, do what we all do. Grab some, dry it, put it in however you store your stuff, and when you get inspiration or someone wants to sequence it, send it to them.
okay. I
Yeah.
guess I'll just keep it in class Agaricomycetes for now. Um,
And unless you do some micro
Yeah,
that
yeah. I mean, I guess I can't rule out some weird ASCO I've never heard of. That texture,
then again,
you
if
know.
you do some microscopy on it and you find some ASCO, that could just be the thing that's growing on it.
Yeah.
That's true too.
I think that would be my opinion, and whatever that is worth. Uh, some sort of polypore that has an infection that makes it really, really, destroys all the, oh gosh, what, skeletal hyphae. So it's not as rigid as normal polypore would be.
okay,
Yeah.
that's
You-
definitely okay. Now I feel better about not knowing what the heck it is. Um,
So the other side, the side that's not black, is the upper side, is it white?
this is the upper side.
Oh, that's the upper side. Okay.
Yeah.
Okay.
So
Yeah.
it's just like a smoother texture. It's
Mm.
got a little bit of rings, and then it's got that like white lip.
Yeah,
Uh-huh.
it
Oh, and
looks
then
like
the
a
underside's also dark. Okay, I
Yeah, the
see.
underside is also dark, but it's like
Yeah.
this like velvety texture and a little bit
Texture.
more of like
Yeah.
purple.
Yeah. Tag Myco Freak.
yeah.
There you go.
See, because he's pretty good at these. Yeah, that's him.
I'll just say I'll just tag him. He can make his own conclusions.
Yeah. He's in the club. He knows.
Okay, well that's all that I have. Um, thanks for all chiming in. Um,
Interesting finds as usual.
yeah. So I'm sure I'll be finding more as the weather warms up and I keep on flipping logs.
Yeah, thank you for sharing.
Yeah, we look forward to it.
Yeah, I'm gonna finally put this that slime mold, that yellow one, a pretty good idea for it in the chat.
Nice. You want to go, Paul?
Yes, thanks. That was very interesting, Jamie. Let's see, share. I think it's this. Maybe. Is that it? That is it. Okay.
It is.
This one. This is, I thought I had a pretty solid ID on this. I'm not, I think this is genus Resupinatus, which I don't have a whole lot of experience with. This is my first picture where I've got some sort of reference in there, my typical dime photo there. So they're pretty small fruiting bodies here. But they're really rather hairy on top. And I did find a species that, in fact, the common name being the hairy oysterling. What did I call it? Trichotis. Resupinatus trichotis. Here's the underside.
Look at that.
Which this is my only underside shot that I've got. It's a little out of focus there, but
It's a good shot. Look at how small they are. That's a great shot.
Yeah, yeah. What do you know what kind of wood that is?
it is. It's a tulip. It's a pretty well rotted tulip. And so I thought this was pretty solid ID. This hairy oysterling, and then I started looking online, and every other example that I could find had this hairy portion up top is always much darker than the whole rest of the fruiting body, and these are just so white. Or maybe they're just old and maybe a little bleached.
Well, I think some of the other possibilities here, Aplanatus, right? Then a couple of the other ones, they also are tomentose.
They are okay.
I
Right.
think I remember trying to work these out a couple of years back, and just it's Resupinatus, and I don't know from there, even with spores and all that. But yeah, you could be correct here.
Yeah. Right.
I think I would probably call that. I mean, look at how tomentose it is, but
Yeah, they look petable.
If only they were larger.
It's
Beautiful,
ridiculous.
really. I've never seen those.
What's the species name proposed here? Because I'm looking at Michael Quebec.
That's something called Resupinatus trichotis.
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 Yeah, I don't think they have that one. Let me see.
So Jamie's saying she finds it a lot on the white cedar.
Yeah, this is definitely on tulip.
yeah, I find them most. I find Resupinatus most often on hardwoods. Well, deciduous trees. Yeah, I don't know. I've tried to work them out. I agree with Resupinatus, but
all right. Let me go check back on some of the other ones and just look for this tomentose situation. These were really pretty impressive. Some of these, it's like some kind of a bleached mohawk look sort of thing. All right.
they also have an interesting shape to them. A lot of Resupinatus. Well, whenever you find them throughout their maturity, they differ, but these are very kind of conical, bell-shaped. Often they're quite flat. So,
okay.
Could we see the underside again real quick?
Sure.
Oh, so they all emanate like right from that stipe spot.
Yes.
Yeah, that's cool.
27 species of Racupinatus listed in INAT, so there's a lot
Oh,
there.
great. Okay. All right. Well, we'll take another look. Maybe I'll reconsider this one here.
Yeah.
Appreciate that.
No, leave it up and let someone disagree with it. That's
Yeah.
Okay. Yes. Okay. We'll see how that goes.
And maybe
I
it's
found
the
a
20.
photo online that looks that's identified as Trichotis, and it looks like this. I think I'm going to put a link in the chat. I think it's the right link. Sometimes this gets confused. It's a gigantic link. I don't know if it's the right one, but you can look at it.
Maybe it's our 28th species too.
But the picture I found has the white hairs like on the
it does.
part of the
Okay.
cap.
Okay. Right. I shall take a look at that for sure. All right. That's the main one I wanted to talk about, but I did have a neat picture since we were talking about the wasp nest slime mold. I did do some micro on this. And this capillicium here, which is so interesting under the microscope that Brandon was talking about. Here's my spores, by the way, which is kind of the best I can do with my microscope. There seems to be some sort of ornamentation there, but
That is pretty good.
Yeah. So this is a good shot. You can see that the spores are granular and they're not reticulate. So those little bumps, they're not connected, which other species in the order they are. And you can see spines and things.
Yeah, okay. Right. And measured those. And but here is this interesting
there it is.
This I think my understanding is sort of a hygroscopic, these strands that are some assist in spore dispersal. But you can see the spiral and spiky nature of it, which is so cool. Got one more where
braided and spiked.
Here's in particular.
Beautiful.
Here,
that's a great photo. Wow.
Photo contest right there.
Oh boy, that's really nice.
I did have to stack a few photos to get this one all in focus, though.
What do you use to do that?
Well, I do. I have a trinocular port in my microscope, so I do have that dedicated digital camera, and then I just sort of tweak the fine focus and get a number of shots attempting to get all the depth of field that I'm trying to get, and then I put it into a program called Helicon Focus, and it just renders it and just merges them all. It's pretty, even I can do it. So, but yeah, that's what those little strands look like, which I was really enjoyed looking at that. So, and I saw
for microscopy, nice microscopy photos, slime molds are a cheat code. They're fascinating under the microscope. They're beautiful.
Yeah. Yes, exactly. And I think this is another one here that you were talking about, Brandon, this oligonema or oligonema. I found this one under a log as well. That's a holly. And this one had similar looking, oh, here's the spores are cool.
Yeah, and you can see, so this is the difference between reticulate and, well, these are reticulate and simply warded. If you can zoom in and you can make out the spines connecting, right?
Yeah.
So they're like ridges.
Okay. Yes, I see that.
Yeah.
And then one of the strands.
Yeah.
Oh, look at the spike at the end of that one.
Go to the end. Yep. That's diagnostic as well. And you can see that this one's not spiked for the most part.
Right. No, really not at all.
Yeah.
Okay. So that's another one. I wonder about how is spore dispersal going on here under the log there? Is it insects that are crawling around and carrying these spores? Or what's going on there? Worms, maybe?
Little bit of everything, right? It's the same question as with fungi. What are these different tools for spore dispersal that these organisms have evolved to? And it's across the board. Yes, it's there are insects that predate the spores and then disperse them in little pockets, like pollinators, kind of. There's wind dispersal. There, with slime molds, it's even more interesting that they can position themselves to fruit where they really want to with their plasmodium.
Oh, right. Mhm.
But again, how do fungi disperse their spores?
Oh,
That's
like
we
feeling
could
for
talk.
the top of the hill or whatever.
Yeah,
Oh
yeah.
man.
But the question of how do fungi disperse their spores, we could talk for hours about that, right?
Yeah, I suppose so. All right. Well, I think that's going to do it for me. Thanks. Let me unshare. How do I do that? There it is. Stop share. Thank you.
Cool
That's
stuff.
fantastic.
Yeah, thank you.
Yeah, some great photos.
Thank you very much.
Does
Oh, you're
anybody?
saving some of these things to send in sometime too, Paul.
Oh boy, that's my next frontier to start saving stuff. And yeah, I guess it's not too onerous of a thing to do. I should really should start doing that. I've got some opportunities with the Mycological Association of Washington. They I think they've got some budget for um, sequencing. So maybe I'll start that this year.
Yeah, do it.
Oh, so uh, do you know last week's speaker?
I know of him. Yeah, I've been on a couple of walks with Tom McCoy, but I think he lives in Pennsylvania, and so I don't know how much he makes it down for some of the walks here,
Ah,
if
Do
I'm
you
not mistaken.
do you attend the Sequanota foray?
I have not yet gone to Sequanota. No.
Yeah, it's really good. It's a
Yeah,
really good foray.
maybe this will be the year. Definitely going to try to get to the nymph foray. Now that you guys turned me on to that, you guys are sponsoring that, aren't you?
Yes, we're
Yeah,
one of
well,
the host clubs for that. Yep.
Host. Sorry, yeah. I'm looking forward to that.
Yeah, it's a really nice area. It should be a lot of fun.
Cool.
Yes, did anybody else have anything to share? I had one or two that I maybe could get some direction on. Let's see. This I have one. This is the side of one of my compost piles. I pulled the sleeve off of it and there was like a little band of them. And I grabbed a couple of them, some which blew away, and I have one more. These things are tiny. The little seed matter you see here, these are like squash seeds. So maybe, you know, 2 centimeters tops on some of them seeds. But there's that one I know like grows in the compost piles.
So no mature ones opened up yet?
Nope, they were all just, just like that. And I mean, it could just be too because like there was plastic like right up against this. So it was like there's probably no kind of movement at all in there.
So what is this, like a Coprinellus or
I don't know. Like the AI was trying to give me a bunch of weird stuff, but
Yeah, that would be my guess. Like Tuacesis or
yeah,
Coprinopsis or
well,
Coprinellus, something along those lines.
yeah, one of the the dung loving ones, right? Aren't there a lot of those that like dung? Is that what this is on? What are we looking at?
I mean, it's like some wet anaerobic compost. So, I mean, dung does not is that's not wrong.
Hmm, hmm.
I get like Coprinellus or Coprinopsis or something on my compost too, so that sounds right.
Cool. Yeah, I'm trying to save this in my new this is my yard herbarium. And then where's the no, no, no.
Is it still out there in your yard, these things?
I was gardening, so I was moving this compost pile.
Oh, okay.
So I grabbed a few, and then a couple of them like blew away from where I had them
Uh,
sitting. So I have one, and then this is on a root of my willow oak, and I was wondering, like, if I wanted to save a chunk or do something, I just grab one of them orange bits, and that's as good as it gets.
To do what? Like
I don't know.
micro?
Yeah, anything.
Is
I
it like
mean,
the canker, the hobnail
Yeah.
canker? I can't really see from this level what we're looking at there. Or are
it,
they
it,
bracket like?
no, no, these are like the canker, I think.
Yeah.
See, and that's an interesting one because I often see what I would call Endothia gyrosa, the orange hobnail canker, on exposed roots. Um, I've never seen one on a exposed willow root, and these look a lot different. They look bigger and more expanded than I've ever seen Endothia gyrosa or one of these, orange or red cankers. So
Expanded.
yeah, yeah, like look at how flat they are at the like 10 o'clock, 10, 11 o'clock.
Hmm. Yeah, I don't know. Cause like this spot is like it gets a bit of traffic, which is why you could see the roots worn down, like right there on the top. So I wonder if I'm just like beating it up.
Mm,
And this is another section of that same root. Yeah, I don't know. Maybe I'll just collect a few of them chunks and poke
Well,
around, see what happens.
what's the size on each one of these like individual fruits before they blend into each other? You know,
Oh, let me get my. Oh, like maybe I don't know, maybe 3 millimeter,
yeah, I guess
if
that's
that.
yeah, I guess that's not all that off.
You ever
Yeah,
look at these? Like, did you ever chop one up or anything?
yeah, yeah. But it's one of those things that I think everyone just calls them. Well, they only Endothia is a new thing. They just called all these things the same for years, and then the chestnut blight happened. They figured out that's a different species that really likes chestnut. So it's only really been since the chestnut blight has been studied that these have been studied. I asked Tom about that last week, and he knows the chestnut blight one. He knows chestnut trees, but who knows willow oak? Who knows the blight of the willow oak?
Yeah. Yeah, I mean, I guess they could all be host specific or whatever. He said that no one's really working on these things. All right. I'll take a chunk and give it a go. All right.
All right. That was fun. I need to stop for tonight, though. However, see you all next week, hopefully.
You have a good evening, Lila.
Thank
Thank
you.
you.
Thanks,
Good night.
Chris.
All
Bye,
right. Good night,
everybody.
everybody.
Night all.
Good night.
Right. Thanks, guys.